So let's get started. We are going to study about the development of female gametophyte. Okay, so the first, female gametophyte is also called embryo sac. We all knew that. Depending on the number of megaspore nuclei taking part in the development, there are three types of embryo sac according to the legend Maheshwari. So there are three types of embryo sac depending on the megaspore nuclei. Those are taking part in the development process. So these are monosporic embryo sac, bisporic and tetrasporic. So first we have to go through the monosporic embryo sac. So let's get started. It is develops from a single megaspore of the spore tetrad, other the three being degenerated. Okay, so in a megaspore mother cell, the four megaspores are there in a spore tetrad, right? So in monosporic embryo sac, what happens? A single megaspore of the spore tetrad is alive, but the three being degenerated in the monosporic embryo sac. Then in bisporic embryo sac, what happens? In this case, the embryo sac arises from a binucleate megaspore of the dyad stage. Okay, so it arises from the binucleate, that is two nuclei in the megaspore of the dyad stage. Okay, then tetrasporic embryo sac. It is develops from all the four haploid megaspore nuclei which have not separated by the wall formation in any of the meiotic division of the megaspore mother cell. Okay, so we know that the megaspore mother cell two meiotic division occurs. After that the four megaspores are formed, right? So in tetrasporic embryo sac, it develops from all the four haploid megaspore nuclei, and these are also not separated by the wall formation, that is cytokinesis process not occur in this tetrasporic embryo sac development stage. And according to Davis, nearly 81% of the angiospermic families exhibit the monosporic embryo sac. Nearly 81% of the angiospermic families. And there is the development of embryo sac involves three identifiable steps. So we can identify the embryo sac's development by the three steps, that a series of mitotic division occurs without the cytokinesis process, and then the cellularization process of nuclei occur. So what does it mean, the cellularization process of the nuclei? The nuclei get cellularized when they form in egg cell, synergid cell, or we can say antipodal cells. It forms after the mature embryo, right? And cell differentiation also. The development of the embryo sac begins with the elongation of functional megaspore, usually that is located at the chalazal end of the tetrad, in the tetrad condition at the chalazal end. So chalazal end, it contains the functional megaspore. After the elongation of functional megaspore, development of embryo sac occurs, right? Small vacuoles appear in its cytoplasm, and the latter fuse to form a large vacuole. So small vacuoles in the cytoplasm, it forms small vacuoles, and latter condition they are fused to form the large vacuole. The megaspore nucleus divide mitotically along the micropolar chalazal axis. So here we can see, if it is an embryo sac forms, right, it is called the micropolar end and it is called chalazal end. We both know that. Okay, so what it says, the megaspore nucleus divide mitotically along the micropolar chalazal axis. In the micropolar and chalazal axis, the megaspore nucleus, the megaspore small megaspore nucleus, they get divide mitotically. means mitotic division. What do you mean by the mitotic division? It forms two daughter nuclei by the first mitotic division, and after the continuous mitotic division we can form two daughter nuclei to four daughter nuclei, 4 to 8, 8 to 16. So it forms at the rate of 2 to the power n, right? So in the mitotic division we can apply this formula to get the right answer, that how many numbers mitotic divisions are there, how many numbers of daughter nuclei are formed, right? So then there is no wall formation, so two daughter nuclei are formed. So wall formation is not occur here. That means cytokinesis process is inhibited here. So we can see it in the diagram. We can see, okay. So there is no wall formation means in the embryo sac one in the micropylar chamber region one nuclei, and in the chalazal chamber one nuclei is present. So these are the two daughter nuclei. These are formed at each pole of the embryo sac, and due to expansion of the vacuole the nuclei are pushed towards the opposite pole of the cell. If the vacuole is present here and it expands to time, and whether so at the time of expansion the two nuclei before the two nuclei reach the micropylar and chalazal region, when the when the vacuole get larger in size to size, the nuclei get and due to pressure the nuclei get separated from each other, and they get the position. The first nuclei get the position at micropylar, and the second nuclei get the position at the chalazal region. Okay, and after that they both the both nuclei divide twice mitotically, is forming two is forming what is 4 nuclei at each pole, and at this stage all the 8 nuclei are found inside the common cytoplasm. So the whole process I can explain you easily. So here after the nuclei get separated to each pole, that is to the micropylar, one nuclei to the micropylar and the other nuclei to the chalazal region. So after that what happens, let's see. Here we can say, so let's assume it is a type of embryo sac. Okay. So there is one nuclei at the micropylar region and the other nuclei at the chalazal region. The first mitotic division occur, and here also first mitotic division occur. So after that two nuclei form, and here also two nuclei form, and it undergoes the another mitotic division, so it can form the 4 nuclei at the micropylar chamber, and here also 4 nuclei formed at the chalazal chamber. So here what happens, let's see, the 3 nuclei get arranged at the micropylar chamber, and here also 3 nuclei are arranged at the chalazal chamber, and the 3 nuclei arranged to form the egg apparatus. What they form? The egg apparatus, and the other 3 nuclei at the chalazal region, they form the antipodal cells. So and the remaining 1 nuclei at the micropylar and at the chalazal chamber, they move towards the central region of the embryo sac, and they are known as the polar nuclei. In some cases, the two polar nuclei get fused to each other and form the 2n polar nucleus. What? They forms the 2n, I mean diploid polar nucleus in the embryo sac, right? So here we can see here After this, the main cell undergoes elongation and takes a sac-like appearance. Cellular organization of the embryo sac then starts. Out of the 4 nuclei towards micropylar side of the sac, 3 get organized to an egg apparatus, and the fourth one remains free in the cytoplasm and is known as the upper polar nucleus. And similarly, the Chalnachal chamber and the 4 nuclei, out of the 4 nuclei, 3 get organized and one is goes to the the central region, that is called the lower polar nucleus. So that's it, the case. And the two polar nuclei gradually lie close to each other and in some cases get fused to form the 2n secondary nucleus. And the two polar nuclei at the central region, they form the 2n, that is diploid secondary nucleus. And this is I forgot one thing to say, that this type of embryo sac development leads to 7-celled 8-nucleated embryo sac formation and is seen commonly in most of the angiospermic families. Okay, it's seen in most of the angiospermic families. That term you have to remember, that 7-celled and 8-nucleated embryo. So right, oh my god, 11-minute long video. I hope you understand all of these stuffs. Thank you very much. If you like it, so then share it the video and subscribe my channels. Thank you.